Hi, my name is Kevin Johnson. I'd like to share with you a new way to model gastrointestinal motility. It's called the multiple moving plug model. I'll compare it to the ACAT model so you can see the differences and why the multiple moving plug model is better. To begin with, I'd like to cover the general nature of both models and their assumptions. And I have here a piece of bicycle inner tube, and I chose this as a prop because it has about the same diameter as the small intestine, about 3 centimeters, and it's flexible like the small intestine. The human small intestine is about 7 meters in length, and because the ACAT model treats the intestine as 7 stationary compartments, each compartment on average would be about 1 meter long. So I cut this piece of tube to 1 meter so that this represents about the average segment or compartment in an ACAT model. Now, one of the assumptions of the ACAT model is that the contents of the compartments are well mixed. So that means that the concentration is the same at every point in this segment, as is the permeability. In reality, though, this wouldn't fit in your GI tract as in your abdominal cavity as it is, so it must be folded to fit. So really, this is a better representation of the ACAT compartment. And remember, the assumption is that this is well mixed. In comparison, the functional unit of the multiple moving plug model is a fluid plug that moves through the GI tract. And this particular plug would represent about 30 mils. So what I want you to imagine is if this were a plug of fluid inside this segment of intestine, it would move through there being propelled by peristaltic contractions. Now, they can be back to back, or they can be spread out also. And as it moves through the intestine, the conditions it encounters change, and so the parameters associated with this plug also can change continuously. The things that can change in addition to permeability are the volume of the plug. It can increase or get smaller. And also the pH inside the plug can change, so you can model the effect of pH on drug dissolution and precipitation. Now, one of the assumptions that's the same about both models is that the functional units are well mixed. So both models assume that the contents are well mixed, except that with the multiple moving plug model, that's a much smaller volume than the ACAT model. And like I said, since the intestine is folded, the pathway that the multiple moving plugs would take would be very tortuous. Let's compare the ACAT model to the multiple moving plug model directly. As I mentioned before, the ACAT model treats the small intestine as a series of 7 stationary compartments. And it's important to remember that these compartments actually represent fairly long segments of the intestine. The barriers shown here are shown as breaks in this model. And this drawing was scaled down from a length of the intestine of 7 meters to a diameter of 3 centimeters. Within each compartment, solid drug particles are dissolving, and both solid and dissolved drug move from compartment to compartment in a first-order manner, independent of the fluid that's in the compartment. Now, there's no physical basis for this model. There are no barriers in this small intestine, and therefore there aren't any compartments. And this model can't be experimentally reproduced in the lab. For example, there's no known barrier that would allow solid drug particles to move across it while retaining the fluid that the particles are suspended in. In contrast, the multiple moving plug model treats the intestine as it is, that is, a long unobstructed tube through which multiple plugs of fluid move due to peristaltic contractions. Magnetic resonance imaging shows that the fluid in the small intestine does indeed exist as discrete packets or plugs. And that's consistent with the multiple moving plug model, but it's inconsistent with the ACAT model, which assumes that the fluid pathway is continuous. Now, the multiple moving plug model assumes that the mass in the plugs moves with the plug. So the only way for mass to exit the plug is for drug to dissolve and to cross the intestinal membrane, as opposed to the ACAT model, which assumes mass moves from compartment to compartment. You'll notice in the multiple moving plug model that there's no transit rate constant between plugs. They're completely independent of each other. Now, both models are going to use a residence time of about 3 to 4 hours. The ACAT model achieves that by inserting numerous compartments to retain mass in the small intestine. In contrast, the multiple moving plug model assumes that the multiple plugs move at such a speed that it takes, on average, 3 to 4 hours for the plugs to move from the beginning of the intestine to the end. Now, the number of plugs and the volume of the plugs and also their timing is based on the actual gastrointestinal physiology, the gastrointestinal motility. And that'll depend on whether the stomach is in the fasted state or the fed state. In the fasted state, the pyloric sphincter will be relaxed. And so when a peristaltic contraction moves through the stomach, there'll be very little resistance to flow through that sphincter. In comparison, in the fed state, that sphincter will be constricted. So when the peristaltic wave moves towards that sphincter, there'll be more retropulsion and mixing as opposed to expulsion of fluid through the sphincter. So you might imagine that in the fasted state, these plugs will be larger in size and there'll be fewer of them because with each contraction, a fairly large amount of fluid is expelled. In comparison, in the fed state, there'll be more plugs and they'll be smaller in volume because of the constriction of the pyloric sphincter. And it'll take more contractions to empty the stomach. Now, there's no inherent first-order assumption about the nature of gastric emptying or motility with the multiple moving plug model compared to the ACAT model, which assumes everything is a first-order process. This model will readily accept imaging data that shows this timing and the volume of the plugs, and it can use that physiological information directly. Now, a big difference between these two models is how they model change. With a multiple moving plug model, everything is modeled from the perspective of the moving plug. All the parameters associated with this plug can change continuously, and they're all completely independent. For change to happen in the ACAT model, mass must move to a new compartment for that change to occur. So all the parameters associated with each compartment are specific to that compartment and will stay the same, where these will change continuously. And you'll notice, for example, if you look at this segment here in the ACAT model, the same segment can hold multiple plugs, and the parameters of any plug can change from the beginning of that segment to the end. So it can change continuously. This gives you more definition in the model for the multiple moving plug model compared to the ACAT model. Now, the time determines the position of the plugs and therefore the environment that it'll be in. And I'll show an example in a minute of how you actually accomplish that. With both models, the total amount of drug absorbed is added up continuously, either for the compartments or these multiple plugs. In the future, look for a new video that will describe how precipitation occurs in the multiple moving plug model. And I also will give you a URL now that will allow you to view actual fluoroscopic videos of gastric emptying and motility in the GI tract. Before I show simulations using the multiple moving plug model, I wanted to share with you the system of coupled differential equations that define the model, and they're shown on this side. Now, this model is a natural extension of the well-established Johnson dissolution model shown on this side, and natural in the sense that the form of the differential equations is the same between the two models. And also there are no new parameters or variables when going from one model to the other. The origin of the Johnson dissolution model is in a paper by Dressman and Fleischer in the Journal of Pharmaceutical Sciences in 1986. And I've kept all the symbols the same as the work of Dressman and Fleischer. And you can also find the derivation of these equations and the definitions in their paper. What you won't find here is the Q flow rate term used by Dressman and Fleischer, because I don't consider, or I don't model motility as the transit of mass between compartments. Now, in 1989, in the International Journal of Pharmaceutics, Hintz and Johnson published a paper that extended the work of Dressman and Fleischer, and that was done to simulate the dissolution of a polydispersed powder. And that was done by introducing an index I to track the individual particle size groups that make up the polydispersed powder. We also considered a time-dependent diffusion layer thickness, so that as the particles dissolve, the diffusion layer thickness would get smaller with the particle size. This model has been studied and cited extensively. In particular, in 2006, Gino and others in the Journal of Controlled Release reproduced this model independently and validated the model with experimental dissolution data. Originally, the model was used to simulate dissolution in a single volume of fluid, V. So this equation describes the rate of change of solid drug in that volume. This is the rate of change of dissolved drug in that volume. And this is the rate of change of drug mass reaching the central compartment unchanged. What the multiple moving plug model does in essence is it subdivides this volume into multiple plugs and introduces a new index J to track those individual plugs as they exit the stomach and move through the small intestine. What you should notice is that in going from the Johnson dissolution model to the multiple moving plug model, one-dimensional array variables become two-dimensional array variables, and parameters that weren't array variables become one-dimensional array variables in the multiple moving plug model. Now, because you have multiple moving plugs now, when you sum up the total amount of mass that reaches the systemic circulation unchanged, you have to consider contributions over all the plugs, and you have to also sum it over all the particle size groups. As I mentioned earlier, we model change by having parameters change continuously depending on the position of the plug in the GI tract. So variables like the volume, the solubility, and the absorption rate constant can change continuously. You could also have the pharmacokinetic parameters change continuously, and you would most likely tie that into a concentration at some location in the body. One of the nice things about having a mechanistic model is it's possible to validate it in the lab. And an example I'll give that is with volume. So if you want to consider volume changing in vivo, you might do it in vitro by having volume change in a fixed way, either through evaporation or adding it. And then you can redo the dissolution with the changing volume and see how well the equations predict the expected change in the dissolution data. To reiterate, the multiple moving plug model is mechanistically compatible with imaging data, which shows that fluid empties the stomach and moves through the intestine as discrete plugs of fluid. These two graphs illustrate a simulation using the multiple moving plug model. For simplicity, I've chosen 4 plugs to do this simulation, and though not accurately reflected in this drawing, I've assumed that all the plugs are equal in volume and contain the same amount of drug. I've also assumed that while the fluid is in the stomach, the absorption rate constant is 0, and when the fluid moves into the intestine, the absorption rate constant increases to 0.07 reciprocal minutes. I've also assumed that the plugs empty at time 0, 15, 30, and 45 minutes. Let's look at one of the plugs, and in particular I'll choose the last plug to exit the stomach, and that's shown in the green profiles in these two graphs. So while that plug is in the stomach for 45 minutes, the absorption rate constant is 0, and therefore there's no absorption from that plug. Starting at 45 minutes, the absorption rate constant transitions from the value of 0 to 0.07, and I've expanded that portion of the graph here so that you can see the shape of the profile over that 1 minute interval. Now you can change this interval and the shape of that profile to anything you like. So you can see that when the plug enters the small intestine, the absorption rate goes up, and therefore you're beginning to see drug in the central compartment due to that plug. The dotted line is a summation of all 4 of these, the absorption from all 4 of these plugs. So you're adding up these 4 curves to get the dotted line. I've also shown a profile here which represents a simulation where all the fluid empties the stomach immediately. So you can see the effect of gastroemptying by, in this particular case, it reduces Cmax and increases Tmax in the peak plasma concentration. Now, I don't think it's realistic to assume that emptying occurs at 15-minute intervals. The reason I spread it out like that is so that you could see these curves very distinctly. The important thing is that you have a mechanistically realistic model that can utilize imaging data for the actual volume and timing of the emptying of these plugs, so that you can do simulations to help you better understand and predict the effect of gastrointestinal motility on absorption. With the multiple moving plug model, researchers can study the effect of gastrointestinal motility on drug absorption using actual physiological data, like the timing and volume of plugs emptying from the stomach. The multiple moving plug model will bring us closer to realizing the full potential of physiologically based pharmacokinetic models for speeding up drug development. To help, I've published a book, Computer Simulation for Pharmaceutical Scientists. This book teaches the computational techniques needed to develop physiologically based pharmacokinetic models. There's a chapter on the multiple moving plug model and the code for an ACAT model so you can compare the two models side by side. Just Google the title and you'll find an Amazon page where you can order the book. If you'd like, you can hire me as a consultant. Please send me an email for more information, and thank you for watching the video.